Okay, I'm going to talk to you today about one short trial that we've done in Australia. We actually started this about 3 years ago. So, when you put this talk that I'm about to give together with all the things you learnt yesterday about how a trial should be done, and how specialist cheesemakers should behave themselves, and how scientists should stamp their feet and make sure that everything's done in triplicate, you'll see all the holes in what we've done. What you will also see, or I hope you'll see just from the fact that I'm willing to stand here and give this talk, is that I thought what we were going to do would probably work. What I was not prepared for was that even though I can't put a single statistical significance on anything, everyone involved in it is convinced it's one of the most amazing things we've ever done. So I hope the enthusiasm rubs off, if nothing else. Oops. Okay, starting at the beginning. I always like to do that. Cheesemaking is 7,000 years old, at least. Its beginnings are lost in the mists of time. Its beginnings of dairying are lost in the mists of time. But that's not true in Australia. We can put a date on it. It was January 1788. I'm not sure if it was January the 25th or 26th. When the first cattle were landed in the new British colony of New South Wales. There were sheep and goats and other things there as well, and chickens and ducks and I don't know what else. But there was an intention to establish a dairy herd of cattle. Huge herd it was too. 3 or 4 cows and 2 bulls survived the trip. So it wasn't much of a beginning. But there was the beginnings of a dairy herd. I gather it was shortly after the King's birthday celebrations in June that year that one of the attendants left the gate open. The entire herd wandered off into the surrounding bushland. They were found 7 years later. I think some other cows arrived in the meantime, but basically it was a fairly shaky start to dairying in Australia. And let's face it, the support they got from head office was not good. And Australia's specialist cheesemakers 200 years later felt basically the same way. They were increasingly frustrated at the simple fact that they were not allowed to make raw milk cheese and that the range of microbial cultures they could get their hands on from the usual suppliers was limited. I mean, I think that's probably a common story everywhere. But I can assure you, in the country that one of our prime ministers once referred to as the arse end of the globe or something to that effect, it has always been true. So the aim of Australia's specialist cheesemakers was to achieve increased microbial diversity in their cheeses. Okay, that makes sense, but how? Whoops, there's 2 ways of doing it. 2 obvious ways to do it. Raw milk cheese, or more interesting cultures. The raw milk cheese thinks that the ice started to thaw this century, and the last changes to the rules were in 2015, and it is now possible to make raw milk cheese in Australia, as long as you do a few things. Obviously you can make cheese with pasteurised milk, but there are restrictions on how you can make cheese that hasn't been, first of all, put through pasteurisation of the milk. Various heat treatments and holding the cheese for a period of time, cooking in the vat, moisture specifications. You can see that that suggests certain microbiological hurdles, but also suggests certain cheese types. But if you seriously want to go for other raw milk cheeses, there are these marvellously named additional requirements. And they revolve, it is a very much abridged list, around animal health and hygiene and feed and milk cooling and using the milk within 24 hours. And if you're doing both raw and pasteurised, keep the milk separate. Have very good process and product composition control and record keeping. It's all terribly sensible. But there are these 2 microbiological criteria that it might be difficult for a small cheesemaker to try to, not necessarily to meet, but to verify and justify that they can meet. The pathogenic organisms in a raw milk cheese must not exceed the level in the milk, and a raw milk cheese must not support the growth of pathogenic organisms. Now, if you can come up with reasonable theoretical or practical grounds to say that this is true, that's fine. But if you can't, You're going to have to do challenge studies with pathogens in cheese and demonstrate survival or not. Really quite onerous and potentially very expensive. So even though the various regulators in the different states of Australia are generally supportive in these matters, it's no surprise that few Australian cheesemakers have actually taken up the making of raw milk cheese. Okay, Sebastian's done a lovely introduction to my talk. I don't have to tell you about cultures. You know all about cultures. You probably knew all about them before he spoke too. But, yes, of course, we use cultures. But what's the problem with cultures? What's wrong with the modern cultures? Why can't you just go and buy a culture and do wonderful things in your cheese? Well, I will say, as somebody who used to work a lot with industrial cheesemakers, there's absolutely nothing wrong with modern cultures. They work well, they're consistent, and you can make good cheese with them. I don't know what you people are complaining about. And then you come into a room like this and then, oh, there's everything wrong with them. They're tamed and simplified, they're not complex enough, and the cheese is boring. Okay, so let's assume, just for the moment, suspend disbelief. Let's just assume that cheese made with raw milk actually tastes better. I could put that in much more elegant terms, but let's say we think that. And let's say we think that the microbial composition of the milk has something to do with it. Well, why can't we make better use of the microflora of raw milk to produce adjunct cultures to put into pasteurized milk to make cheese? To at least capture some of the features of raw milk cheese in a pasteurized milk cheese. Now, in a regulatory environment like Australia's, where they try to do things in a fairly conservative manner, this is a pretty appealing idea. So, our goal, apply modern culture technology, define strains, know exactly what you've got, propagate it in a safe and reproducible way. But this allows the cheesemakers to reintroduce at least some microbial complexity into their pasteurized milk cheeses. And more than that, if you isolate it from, say, a single farm's milk, you've got a site-specific snatch of the microflora. It's not everything, but it's a grab, it's something. And you can put some sort of local character into your pasteurized milk cheese. You can then all go down the pub and have a fight over whether you want to call them indigenous or autochthonous cultures, depending whether your classical education is Greek or Roman in its leanings. I like autochthonous. So we were seeking to demonstrate that you could apply some modern science and the modern way of doing things in this cause to educate cheesemakers into that possibility, consumers, and to just put a few ideas in front of the regulators to see how they would react. And I'll say before we started, they were mainly going, oh, that's very interesting. We wanted to assemble some prototype cultures, assess the technical feasibility and costs of making simple cultures, and obviously make some cheese and see how it turned out. So there's the flow chart. Take some milk, have a look at what's in it, isolate some organisms, consider whether they're safe, make some blends, grow them up, provide them to the cheesemakers, taste them, and then work out where to go from there. The participants, obviously the Australian Specialist Cheesemakers Association, represented by Alison Lansley over there. Sutton Grange Organic Farm, very much the movers and shakers behind parts of this work. Prom Country Cheese, L'artisan Cheese, you'll notice we've got goats, we've got sheep, we've got cows. And most of the culture assembly work, in fact all of the assembly work, was done at Dairy Innovation Australia, which at the time was a developer and supplier of defined strain starter cultures and adjuncts to the big manufacturers in Australia. So the first question, are the microfloras of the milks from the different sites, and some of them are a couple of 100 miles apart, genuinely different. So we used 16S ribosomal RNA gene sequencing. Hey, you've heard of that before. So that, we're not doing anything particularly unusual. This diagram is a slightly different representation, but 3 different colours for the 3 different milks. You can see the different peaks representing different organisms. The patterns are different. Goodness me, it's cow milk, sheep milk, and goat milk from 3 farms 200 miles apart. If we couldn't find some differences, there would be something wrong. But we just wanted to demonstrate, you can do this sort of stuff. And if you zoom in and look at some of the slightly more prominent components, you can see there's quite a bit of heterogeneity. However, are these things genuinely different? Oh yes, oh maybe. One point that comes across in this sort of analysis is that most of the organisms that show up in the analysis are things which don't show up in cheese. And you wonder, how hard do you have to dig into the data to really get information on what relevant organisms are there in there? And the other, we used a commercial service provider, which will remain nameless, and we sent them duplicate samples of the same milks, and the answers that came back were completely different. So all we're willing to say at this point is this great technology, it's a wonderful research tool, but for specialist cheesemakers to sort of plug and play the technology and get something out of it, there are a few traps. But we had a look at that and we were reasonably happy with it. We've got heterogeneity. So then we isolated bacteria, and I have to say at this point, how many bacteria? Not many. How many species in a raw milk? What was the figure that was bandied about this morning? More than 300? Okay, we isolated bacteria. We did not look at yeasts and molds. We looked at only one bulk milk sample from each site. We only grew them on one agar medium, brain-heart infusion agar. 30 degrees, 37 degrees, aerobic, anaerobic, but not exactly a huge bunch of conditions. And we picked colonies that looked different, but if they looked like bacillus, we didn't pick them. Now, there's enough biases in there to write a book just on experimental bias. But the idea was we wanted things that genuinely came from that milk and which we thought at least wouldn't be spoilage organisms. 168 isolates were classified, at least to genus level. But which of those isolates would you actually deliberately put into your milk? It's an interesting question, probably more philosophical, or is it legal? I'm not quite sure. There are things which, if they're in your raw milk, you'd be quite happy to have in your cheese. But if I gave you a bucket of it and said, tip that in your vat, would you do it? I've never heard of that organism. What is it? We'll talk about some of the organisms. But first of all, we'll talk about what we cared about, and it was safety. That was the thing we had most in our minds. We didn't want to upset the regulators, and we didn't want to kill anybody. So we were quite cautious in our choice of organisms to include. Obviously, some microorganisms have a long history of use, but some don't. So we scanned the literature, worked out what we thought might be a bit dangerous, what we thought might cause spoilage, any other observations that helped us to decide which organisms to include and which ones not to. 6 of the isolates we have genome sequences of, and we're gradually working through them to see if that tells us anything about their properties. But from those 3 milks, we had more than 40 different species, and I'm not going to read them to you, but if you want to scan through it, you can see some are pretty familiar lactic acid bacteria. Some are not so familiar. Some of them have names which could frighten the horses. So when we drew the line through the ones we weren't terribly happy with, we were left with 15 species. Now that sounds like a fairly savage cull, but it meant that we ended up with adjunct cultures that looked like this. Now, if you scan that list of species names, I've never seen a commercial culture that looks anything like that. Okay, it's a bit simple compared with what's in a raw milk sample, but as a culture to actually prepare in a laboratory and put into a cheese, that's something different. So that's what we got from one site, this from another. You can see this one has some lactic acid bacteria in there. This one, a bit simpler, and because we decided that we may as well be brave, we prepared one culture with 24 different isolates in there. Now, we embraced the fact that strain differences could make a difference. So in some of the cultures where we had a lot of isolates of one particular species, we put in more than one isolate just in case they were different. So we then went from the laboratory to the cheesemakers and said, what do you think of these? And they said, yes, okay. So we grew them in a really complicated way in a bottle of milk with a bit of yeast extract and glucose to help them along. We deliberately kept it low-tech. And then we froze them in small volumes, calculated so that in the vat sizes that were being used by the participants, they'd get somewhere between 10,000 and 100,000 bacterial cells per milliliter when they added it into the vat. So, now we come to the cheese-making trial part. Ho, ho, ho. I suppose we should have been more forceful. Could you try making multiple batches, preferably on the same day, with and without the adjunct culture, and mature them together and have them ready for our tasting day, which will be on this date? Yeah, all right, we've got some cheeses. I won't swear to a perfectly balanced experimental design. But we did have some cheeses made with and without. Do these cultures actually have any effect? We did a bit of instrumental analysis, gas chromatography, mass spectroscopy, looking at peaks of this and that. You could see a few differences, probably more easily seen in this sort of diagram. But really, the question is, what do they taste like? So these were the cheeses that were part of our formal tasting. We had some other cheesemakers that we gave some of the cultures to try out as well. But we had sheep, goat, cow, soft, semi-soft, mold-ripened, wash-ripened, so everything other than cheddar, really. And then we put them to a blind tasting with experienced assessors. And the two questions: did they taste different? Did they taste any better? And this sounds a bit weak, but in almost all cases, the flavours were different, and in most cases, the flavours were improved. And then, I suppose you could say we were digging for excuses, but the ones where there were no difference, well, the cheese was only about a week and a half old. None of the cheeses had any flavour. It didn't make any difference. The ones where it wasn't improved, it was a comparison of blue cheeses that were already quite complex in their flavour, and the experts said, well, it's too complicated. I don't like them. They're not good. They're not typical. So certainly they were making a difference, and mostly it seemed to be a good difference. And then we thought, let's be brave. A meeting of members of the Australian Specialist Cheesemakers Association, so this is a mix of cheesemakers and enthusiasts. The Rootstock Food and Wine Festival, and up in Sydney. You can't trust those people in Sydney. But again, another group of people where we just gave them the cheese. And then we took them to the Australian Society for Microbiology Christmas party. Same approach in each case. It was very informal, and it depended what cheeses we had available at the time, so there's not a lot of consistency to it. But it was, here's a table, here are cheeses. You don't know which ones have the adjuncts in there. Here are 3 cups. Take a token, drop it in the cup of whichever one you like the most. Let's keep it simple. And it was amazing. The blue is the control, the other colours are the ones with the adjuncts. In every case, except this one, quite spectacular preference for the ones with the adjuncts. And we worked through this one, and what we realised was that this cheese was past its use-by date. By the time we'd gone to this tasting and that tasting and waited for this event and everything else, the cheese was going downhill. And the addition of the adjunct accelerated that. So, not surprisingly, you have a culture that actually does something. There are pros and cons to what it does. And this blue. The judges didn't like that one. The public did. We were quite happy with that. So, what have we done? I think we've actually gone back to where culture technology was 100 years ago, applied a few modern tweaks, But instead of trying to get the simplest possible culture, we've gone for something that's a bit more complicated. We've made some blends, which I think are unlike most of your average cultures. And we've made some cheese and we've tasted it, and it's very, very encouraging. So we did our demonstration of the potential application of the principles. We raised some awareness amongst cheesemakers, consumers and regulators, and not too many eyebrows were raised. It was rather good. We assessed the technical feasibility. We grew them in a bottle. Not that hard. And the costs, well, at full commercial costs, we worked out that the development of these cultures cost about 15,000 Australian dollars per culture. So that's what, 8,000 pounds, 10,000 euros. And the trial cheeses, well, we think they were worthwhile. So where do we get to? Next steps. This is the sticking point. Now we start asking ourselves, we did it this way. If you were going to do it again, is that the way you would do it? Would you start with a single animal raw milk, the factory bulk raw milk, or would you actually make a raw milk cheese and isolate bacteria from that because they'd better reflect what would be growing in the cheese? Would you do it by farm? Or to spread the cost, would you do it by region? So you could claim you had a regional culture, which in some way at least represented the microflora of that region. How much variation are we really looking for? Are we driven by enhanced flavour, or are we driven by a desire for uniqueness and regionality? What's the driver behind this? And who could do it, and what would it cost? Could you put some agar plates in front of a farmer and say, here you go, put some things on that, and then, how much could farmers and cheesemakers do, and how much would you really want to trust to a specialist laboratory? And then looking after those cultures in the long term and propagating them, who would do that and what would it cost? Not helped by the fact that Dairy Innovation Australia, that did the initial work, is now out of business. So lots of people were involved. Personally, do you notice I finished a bit up in the air? Because that's where we are. Lots of people involved. I'd personally like to thank Christopher Pillage, who did a lot of the hands-on work in the culture development. And we're open for questions.